Okay, so coming to this chapter, protoplast, isolation and culture. Protoplast is what? It is a nucleus plus cytoplasm. Okay, nucleus plus cytoplasm. That is your protoplast. So means this is the cytoplasm, and then these are the organelles. Okay, this is the nuclear material with all the DNA and all, and the cell membrane and the cell wall, they are not there. Okay, so cell membrane and cell wall, they are absent. Only the protoplast is left. Okay, so we can isolate protoplast in two methods: mechanical and enzymatic methods. Okay, mechanical method, we don't do it anymore because this was done in the ancient, by the old scientists. Okay, it is very straining process. You have to, you have to be you have a very sharp blade or knife, very fine needle. Then you strain your eye in the microscope and you try and cut. Imagine to cut a cell. Okay, so we don't do this mechanical method anymore. Okay, it was just before when there was tissue culture techniques were not there. Okay, let me just say it again. In the mechanical isolation, the old scientists know they will wash those leaves, peel off the epidermis, and then try and cut with the blade. Okay, they try to cut the cell wall with the blade so that they can fuse them. Okay, but we are using the scientific way. Okay, because the old historical way, you can damage the cell. No, you might push the blade too hard and damage the nucleus. You might slip, your finger might slip, and then you might pierce the cytoplasm. Okay, and there may be so many cross contamination. So mechanical is not done anymore. Then looking at the enzymatic method, there are 4 steps. Okay, first you sterilize the leaves, then you peel the lower epidermis, then you have to put it inside the enzymatic solution. It will just melt off the membranes. Okay, then you have to isolate it, and then you have to clean the protoplast. Okay, so you have to take healthy leaves. Okay, so you choose the healthy and the green leaves. Okay, so choosing the material also is important. Then you have to sterilize them. Okay, sterilization, you already know how to sterilize. Okay, I've explained so many times. Then you have to cut it into small pieces after the sterilization. Okay, so what the scientists do is they usually pick the plants that they've already grown in the in vitro method. Means they pick the plants that they have grown, they have cultured in the lab only, because those plants are already in the sterile condition, no, inside a test tube. You don't have to wash and all, no, and you know exactly that there is no contamination, there's no fungus, bacteria at all. So they prefer to take the leaves of in vitro cultured plants. You understand? Those plants that were grown by plant tissue culture method. Okay, because they were already grown in a sterile, aseptic environment. Okay. So you need just a little bit of material, 1 gram or less than 1 gram, okay, to get the protoplast. So the enzymes that you use to dissolve those walls, those are the list of enzymes. I'm not going to read, no point, no, you have to see it yourself, some pectinase, some cellulase enzymes, etc. Okay, so you can prepare a combination of those enzymes in the concentration of this much %. Okay, and here this is the enzyme combination. Okay, a different enzyme combination for different plants. So you can sterilize this solution by cold sterilization, and the leaf or the tissues are placed in Petri plates that contain the tissue and the enzyme mixture. Okay, then you have to seal, and then you leave it for a certain amount of time, 4 to 12 hours on a rocking shaker. Okay, shaker, so that that enzyme solution and those plant tissues, no, they mix very well. Okay, because the cells are very small, no. It's not like just me, I'll just go inside a bathtub and my whole body will be covered with the water. No, the cells are very small, so you have to keep on a shaker. Okay, a shaker is like it's like a machine, no? It looks something like this, and then there are grooves, there are holes for you to put your conical flask in. Okay, then when you plug in this machine, you turn it on, it will keep on shaking. Okay. So after you incubate, means you kept the enzyme and the tissues, then you have to filter it, okay, to remove the enzyme, okay, and to capture the tissue or the protoplast through a nylon mesh, okay, there. Then you have to centrifuge, okay, to remove the debris, okay, to remove unwanted things, you have to centrifuge. The protoplast will form a pellet while the debris and supernatant is removed, okay. The protoplast pellet means it will come down, okay, it will be down here, and then all the rest, the watery one, the supernatant, you just throw it. Okay. So now you have to clean the protoplast, because yes, you have thrown away the supernatant, but there are some debris that have clung, right? So you have to further you have to sterilize it in this sorbitol solution, means without the enzyme. Okay, then you have to clean your protoplast. Okay. Floating protoplasts are carefully removed with the help of sterilized pipette and bulked together for further use. So there are protoplasts that don't form a supernatant, means they are too light. No, they are too light, so they are at the top and then the debris is below and then the junk is down. So you carefully you have to take out this one, okay. So let's look at the method. So okay, so we start with the leaf. They have peeled the lower epidermis. They have sterilized it. Okay. Then they have plasmolized it. Okay. You plasmolize the cells because you want the cytoplasm and the nucleus to be away from the cell wall and cell membrane. Okay. Then you release the cells and then you wash by centrifugation. Okay. This one, in this step, you added the enzyme. Okay, let me just repeat. You cut the cell. Okay, you cut the epidermis. Then you add the enzymes. Then the cells will become plasmolized. Then you wash and centrifuge. Now you have got your protoplast. Then you will culture your protoplast. So you put them on a plate or a petri plate of media, then you culture your protoplast, then the cell wall will regenerate and then you can form a callus or form the plantlets of your choice. Okay, then here the organs have regenerated. We did no organogenesis, then again a normal plant will regenerate. So it's very easy. Once you keep listening to the lectures, no, it's very easy because it's all the method is basically the same, just the techniques may change here and there some steps. Okay, so when you are culturing the protoplast, you have to protect this protoplast, okay, because they are not having any wall to protect them. So you have to protect them in the culture medium until they generate a wall that can protect itself. Okay. So by doing, how do you protect it? You have to maintain the osmotic concentration. Okay. So osmolarity in the medium should be adjusted to the same level as in the enzymes and washing solution. Okay. So not just the concentration of enzyme and the washing solution, but you have to check the osmotic potential. Okay. So you can keep on reducing the osmotic potential as the cell wall keeps regenerating. Okay. So if you decrease it too abruptly also, it will burst. Okay. It will burst the protoplast and it will obviously damage them. So these are some electron micrographs showing you the protoplasts. Okay. So the medium that they use is the MS or the B5 medium. MS is the MS media that I've been talking to you about. Okay, so let's look at how they fuse the protoplast. So in nature, gametic fusion forms a zygote. Okay, then after the zygote form, it becomes a diploid, right? So sexual crossing is the major source of genetic variability. Okay, but if you want to create some hybrid, you cannot just depend on nature, no? Because there will be self-incompatibility. You must have read, no? There may be self-incompatibility. It may be, lethal genes may produce, etc., etc. But if you want to create something beautiful, okay, like a blue orchid, they want to create a rainbow orchid. So they perform some sort of experiments. So there was not much success, no, to interbreed and create hybrids, no. So that's why we go for the protoplast fusion. Okay, it provides you a new approach towards overcoming the incompatibility. Okay. So when you fuse the somatic cells, okay, means the non-gamete cells, to produce hybrid, we call it as somatic fusion or somatic hybridization. Oh, I'm dead. So to fuse a protoplast of two different hybrids, directly you just fuse one protoplast. Let's say this is species 1. Okay, protoplast, cytoplasm and nucleus, and this is species 2. Okay, directly you just contact them, and then suppose this is 1 and this is 2, directly you put them together, then slowly they will travel, no, the cytoplasm will travel, and then after the cytoplasms they merge, then the nucleus will start merging. Okay, then when the nucleus will merge, it will become a dikaryon, no. Instead of 2n to n, it will be 4n, something like that. So how to make those protoplasts attach to each other? Yeah, you can stick one protoplast physically, but they will repel each other, no? It's like magnets, right? They will repel because they are not the same. So you have to do it by electric charge, okay? I don't know where it's written. It must be written here somewhere. Okay, there. So you have to fuse those protoplasts by the electric charge, okay? So the charge from this much to this much voltage, okay? And it can be reduced by presence of calcium ion. So the units you have to remember, okay? Minus 10 millivolt to minus 50 millivolt. So there have been success in many dicots and many monocots, and even plants and even insects also we have fused, okay? Even between a diploid and a haploid also we have fused. So the density of the protoplasts you can determine by hemocytometer. You have done that in first semester. I mean fourth semester or something, no? So you can use some chemicals and other physical agents also to enhance fusion, okay? Known as fusion agents, okay? So these are some chemicals that you can enhance the fusion of the protoplast. Okay, so after you have fused the protoplast, okay, again you will have to okay, let me read this line, okay. After you have isolated each protoplast, you can suspend it in a mixture of chemicals known as aggregation mixture, okay, and you keep it at this temperature for this time, and then you centrifuge it, okay. Then you transfer it to a water bath for this temperature and this minutes, and then most of the protoplasts will undergo fusion. Okay, so this mixture of chemicals and centrifuging and temperature maintaining, no, they will induce fusion. Okay, so this is a diagram how they did it with the electric charge. This is another diagram how they did it, you know, just to demonstrate how it happens if you use chemicals. And this is the overall process. So thank you.